COVID was kind of our wake-up call, our refresher course on pandemics. Whether it was engineered or not is irrelevant to me, other than the fact that we can't easily identify whether it was engineered or not. But it's certainly getting easier to go and make these agents that could have, if designed to be malicious, could cause huge harm or death, not just to humans, but to any species we target, whether it's an agricultural crop or animal or any species. This to me is my biggest fear. With things like longevity, people really interested in how do I live longer? And I tell them, look, based on what I'm seeing and how slow it takes to take a drug and all the research that we have to do around the aging process, if you've got gray hairs like me, you're probably not going to benefit from most of the aging treatments, no matter how much money you have. But if you understand that cloning is not that hard, we do it now for 30, 35 mammals. We clone horses and pigs and dogs and cats. There's no technical barrier to being cloned. The thing I say is, why don't you prepare to be cloned in the future when it is safe and legal? Cloning in general is used in agriculture, it's used in microbiology, it's used in all sorts of cell work. We are going to use cloning as a conservation tool for, to deal with the species that are threatened and endangered, to introduce more diversity into the gene pool, etc. Cloning technology is only going to improve over time. I think it'll be the first human clone that will really catalyze interest in cloning and really get people to demystify it and just see it as another tool in our tool chest.
Thank you for joining Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Mr. Andrew Hessel, who's a pioneer in synthetic biology, recognized for his works as a microbiologist, geneticist, inventor, entrepreneur, and futurist. Andrew is celebrated for his vision of programming life itself, seeing cells as living computers and DNA as a code we can now write, not just read. He's the co-founder and chairman of Genome Project, an initiative to design and build synthetic genomes, founder of Human Genomics, which engineers custom synthetic viruses to target cancer. At Autodesk, Andrew helped shape open source biology to personalize 3D printed medicine. He's a sought-after speaker, advisor, and the co-author of the acclaimed book, The Genesis Machine, which explores the promise and the ethical challenges of rewriting life in the age of synthetic biology. Really, really appreciating time and being part of the podcast. Would be great if you could start with explaining the difference between DNA and RNA, and also explain why DNA is often referred to as the source code of life.
Yeah, happy to do that. So there are two main information encoding molecules in a cell, RNA and DNA. And the way to look at DNA is it's kind of like if you go into a library, they have reference books that they don't let you take out. They're too valuable, so they keep them kind of behind a counter, but you can borrow them when you need them to go look at. That's kind of like DNA. It's kind of like the archive and reference for all the instructions necessary to run a particular cell. And just about every cell in a multicellular organism has its own individual copy of that DNA. RNA is kind of like the photocopy you would take from that reference book so that you can use that information at home or at your workplace. You would go and photocopy it so you could take it out of the library. I realize these are old physical references, not download and stream, but it's kind of this, it's a good analogy, because the photocopies that you would make at a library of the reference material don't last very long. You don't take pristine care of them like you would a reference book. You can get them, you can spill coffee on them, you can get them dirty. They don't last long. Once you're finished with them, you can put them into the garbage. So that working copy of the information needed to run a cell is what's used by the cell to go and make proteins. It's how we send most of the internal messages inside the cell to the various constructive and destructive equipment in the cell. It's all run by the mRNA. Chemically, they're very similar, except that with DNA, you have a slight chemical change, very small. So I hope that's I hope that's helpful. It's a functional description, not a molecular one.
Right. I appreciate you doing that, Andrew. Now, in the 1990s, we got into this Human Genome Project, and the vision was to kind of map out the entire DNA sequence of a human being, 3 billion base pairs of DNA. And around 2003, we kind of managed to sequence the entire genome of a human being. And the promise of that was, that maybe, hopefully, we would be able to, once we are able to kind of understand the entire human genome sequence, we would be able to maybe eliminate all diseases. Why do you think that hasn't happened? Why do you think that promise hasn't been fully realized, even after completely understanding the DNA?
Well, look at it from a different perspective. First of all, our genome is code that has been evolving for 4 billion years, basically since the early ancestor cell. We call that LUCA. Last universal common ancestor, because all life is connected. It's not like you kind of get million-year gaps between life forms. It's all connected. Reading our genome was a huge technical exercise. And there's only 4 letters, well, represented by 4 letters, A, T, G, and C, but there's no instruction manual attached to that. There's no annotation. There's no metadata. We had to go, so reading it is one thing, understanding it is a completely different process. And really, it's been 22 years, like a blink of an eye. So we are making huge progress in understanding. And first of all, reading the code isn't going to help you cure a disease. It's going to help you diagnose a genetically based disease. And we've made huge progress on that front in diagnostics. And of course, the cost of sequencing has dropped by 1 millionfold. So today, a human genome, the first one, which took, literally 13 years start to finish and cost somewhere on the order of $3 billion. Who really knows when there's that much money flying around? It can now be done for a few $100s with all the annotation in less than a day. So I think we've made some pretty good progress. But to your point, if you're interested in curing disease, okay, you have to take that diagnostic information, you have to understand it, and you have to have tools to manipulate. And this is what's really been blossoming over the last 15 years with tools like CRISPR for gene editing. So we can do genetic surgery on an individual's genetic code. Things like mRNA synthesis, where we can actually go and make new instructions, write them out with precision for the cell. And my particular interest, which is synthetic genomics, where we actually learn how to write and assemble DNA code to actually make an entirely new cell design. Starting with just copying the ones that exist, of course, and learning how to edit and recode, but moving us to a place where I believe in the next 5 to 10 years, we'll have the ability to code just about any single cell organism or component thereof. Like, we're on a really exciting cusp here in biology and biotechnology.
How awesome is that? From 1990s to, now we have able to democratize human genome sequencing. And you're saying maybe a few $100, we can actually map up the entire human genome. So how cool is that? And you said that right now, it's not just understanding, it's about also writing. And through the tools such as CRISPR-Cas9, we are actually understanding that. And we are able to do that, read, write, edit these DNAs. And because of which, we have these certain gene therapy, like for sickle cell treatment, then beta thalassemia, etc. But these are way, way, way expensive. When do you think gene therapies are going to be truly affordable and accessible? That's my first question. And if DNA is the programming code of life, why is it still so challenging to program a disease-free human?
That's first of all, we're starting with humans. When we are the very apex organism on this planet, arguably the most complex organism. So, to put it in perspective, we only made the first simplest organism, the bacterium, and even then, of the bacteria, one with the smallest amount of DNA. We only did that 15 years ago. That was Craig Venter and his team making the first fully engineered genome, which was then, I'm sorry to use computer terms, booted up to make a living cell. That's only 15 years to do the simplest. And yet everyone immediately jumps to human at the very top and say, hey, why haven't you cured every disease in humanity yet? Give us a bit of time. That being said, gene therapies are being made. They have been made since I think the first presentation I went to was to cure something called SCID, severe combined immunodeficiency, where people are born literally without an immune system. And if you don't have an immune system, anything will kill you. Like basically, but they were doing gene therapies and that in the 1990s. Today, there's multiple gene therapies for hearing loss, for blindness. Again, more and more are coming out. There was just a recent gene therapy here in the US with baby KJ, who literally was diagnosed very early, and within a small number of months had a custom gene therapy designed, manufactured, and delivered, which was huge. So we're making progress. The ones that do exist today are very expensive because the companies and researchers that are driving the forefront of this are pioneering. They're pioneering. And companies, of course, are also trying to maximize their return. That's what companies do. So they're expensive today. But as this ability to make gene therapies, which truly is not that hard when you start to gain experience and use the software-based tools, automation, and processes that are coming into digital biology. As more competition comes in, I expect to see the costs plummet because engineering biology is extremely inexpensive when you can start to code it. Think of the most cost-effective way to manufacture something complex is to grow it, where it literally self-assembles. Put a seed in the ground and you end up with a tree or a plant. That is remarkable. And with bacteria, the organisms I worked with the most, you can start with a single bacterium that you've made an edit on, a change to, and you will have trillions of bacteria the next day. So the costs will fall faster than any other technology as we start to put the right foundation in it. The thing is, we are impatient, and rightfully so. And yes, there are some true economic costs. And when it comes to things like therapies, human therapies, you're trading off the cost and access to a leading edge therapy, one that might represent a cure. And literally you're weighing the access and cost of that against a human life. So that doesn't happen if you're making a toaster or a cell phone or an air fryer. This is unique to biomedicine. But it's becoming so much easier. It's becoming so much faster. And with the AI and automation coming in, that's just going to keep accelerating.
Right. I hope that this convergence of AI with biotech accelerates this whole tech stack and brings these from the lab to the market. I was on Medium and I was reading an article about one of your articles which says human clone number one. Now you mentioned over there that Dolly the sheep you cloned or was cloned 30 years ago, and then it's not moved forward from there. And you've been one of those pioneers who has been outspoken about the concept of programming life itself from scratch. Can you elaborate this article that you wrote, human clone number one? Where does that stand at this point in time? And when do you think, and what would it take for it to actually come to fruition?
So I'm really fortunate that I work independently. It's been how many years now? 7 years since I've been inside a regular company, the last one being Autodesk, which was an amazing run for me. I love that company. And then before Autodesk, I was with Singularity University, which really pushed everyone to the edge of technology. It was a wonderful community, still connected to that. And before that, I'd spent 10 years exploring this emerging world of synthetic biology. And the term that I use today is digital biology more, because it's not just about synthesizing. It's just much more comprehensive. Biology across the board is digitizing, but so I can speak freely on things that many scientists or people in industry cannot. One of my interests ever since I was a kid was cloning. And that's because of, this series of books, Dune, and classic science fiction. But I read it when I was like 7 or 8 years old. And in the second book, this one, Dune Messiah, one of the characters is cloned and comes back. The character's name is Duncan Idaho, and they call it a Gola in this book. But when Duncan comes back, all sorts of hijinks ensue. But if you read the whole series of Dune books, a classic by Herbert, Duncan is the only character that connects over time because he's serially cloned and resurrected. So that was burned into my brain at a very early age. And I thought, wow, this is so much better than just dying and being buried, or dying and being cremated, or dying and being put on a funeral pyre in the Ganges. Whatever the other options for dying is, resurrection sounds pretty good. And of course, those books were written in the 1960s and 1970s for the most part. But in the 90s, this wasn't science fiction anymore. With Dolly, it became real. And since Dolly, there's been several instances that I can remember of someone saying, we have cloned a human being. And it's caused all sorts of, stories to be told in the press, and everyone gets all upset and everyone starts talking about it. Nothing has been verified to this date, but I go and speak at conferences. With things like longevity, people really interested in how do I live longer? And I tell them, look, based on what I'm seeing and how slow it takes to take a drug and all the research that we have to do around the aging process, if you've got gray hairs like me, you're probably not going to benefit from most of the aging treatments, no matter how much money you have. But if you understand that cloning is not that hard, we do it now for 30, 35 mammals. We clone horses and pigs and dogs and cats. And, there's no technical barrier to being cloned. The thing I say is, why don't you prepare to be cloned in the future when it is safe and legal? And that is not hard. You bank your biology, obviously, in cryogenic storage. You bank some money so your clone isn't bone poor. Why do that? Compound interest works really well there. And then you also can bank information about your life story, history, essentially your digital footprint. And you package that up for your future clone. And remember, clones are just identical twins. That's it. There's no genetic modification. It's just using an existing program and making a new embryo. And so you're still born a baby. You're going to be born at a different time. You're going to have a different mom. I write about all this stuff because there's so much misinformation around cloning because of Hollywood movies. But it's really a practice that's done every day. And in one of these conferences, when I was talking about this, a young man came up to me and said, I would really like to be the first clone. And over a period of 6 months talking with him, he's logical. He can afford it. I just put that article out there because it takes essentially the resources of a IVF clinic, an in vitro fertilization clinic, assisted reproduction, to do cloning. It's not hard, but it takes essentially the resources of an IVF clinic and perhaps some really, an embryologist team. But there needs to be someone that's willing to move forward, in a zone where it's permissive, not necessarily legal, but there's nothing on the books where it's illegal. So by writing a story and putting it out there and doing podcasts like this, maybe I'll get a phone call the next day saying, yeah, we're in. And I can vet that particular group. And then if everything works, if everything passes my sniff test, I can do an introduction to this particular client. And who knows, maybe 9 months later, we will have the first human clone, which, by the way, will be a media storm, but no big deal. Like, we use IVF today. That was controversial in the 1970s. Today, there are something we don't count, but there's something like 14 million kids born by IVF. It's no big deal, including 2 of my own.
Right. Yeah. So I mean, more power to you and your vision. I guess, when you're tinkering around with the deep future, there is, the normal public or the religious folks, they get a little negative about the conversation because they think that, you're tinkering around with nature. But I guess like you rightfully pointed out, IVF, at one point in time, it was not really it was looked down up. But today, there's millions of IVF kids. And I guess maybe in the future, cloning could be something like that.
I think it'll just be a menu choice at IVF clinics. Once we kind of break the seal on something and we understand it, and it's not magic, it's just something you can order online, then people become a lot more comfortable with it. And then this certainly happened with the first, IVF baby, the first so-called test tube baby. There was a media storm because it was brilliant science and medicine, but then they quickly realized it's just a kid. It's a big deal. It's just a kid. And the same thing with cloning. Not only is it just a kid, except that now the history of the genetic program, the first instance of that program. Now it's a kid who's actually being born on a foundation of information and resources and intention that is extraordinary. So I'm looking forward to the first verified human clone. Maybe they exist today already and they just haven't come forward because they're not comfortable and they don't want the attention, because there's no technical barrier. But again, if there's a clinic out there that would really like to do this and feels and is willing to shoulder the risks and the challenges, again, with full transparency, I've got someone, kind of vetted for that process.
I wish you the very best for this. If it happens, obviously it'll be like a landmark. Now, how different is cloning from what maybe George Church is doing with Colossal Bioscience, where he's reviving these extinct species? Like, he was also in the news for bringing back direwolf, and the thought is to kind of, bring back the woolly mammoth and the Tasmanian tiger. How different is this from the cloning technology? I'm just kind of pushing this, because, you've seen movies such as Jurassic Park, because you kind of tinkering around with this, and eventually you kind of want to bring back the species, and then maybe you crossbreed, and then you have something like a T-Rex or something like. Is that something which kind of is back of the head and kind of worries you?
No, it doesn't worry me personally. First of all, Jurassic Park, brilliant, and if we could clone dinosaurs, we'd want to see dinosaurs. Like, there would be a Jurassic Park. Everything else is just dramatic effect. So I'm not worried about that. In general, these de-extinction efforts being done by Colossal and other groups like Revive and Restore are looking at these tools, which are, first of all, you need control over the reproduction of the animal or a similar animal. And you need the ability, which is essentially cloning, or at least assisted reproduction. And then you're largely doing editing on that existing organism to kind of rewind it in some ways to make it, or add in features and characteristics of the original species. That's certainly what they did with the direwolf. That was the plan with the woolly mammoth. You're starting with the base of an Asian elephant. But cloning itself is a little different, whereas you're making an exact copy using pristine genetic material. It's not an edit. In fact, you don't have to do any editing of the genetic material at all. It's a pretty straightforward process to clone when you have access to intact nuclei, essentially the hard drive of a cell, and cells that you can manipulate and have eggs, essentially. So I think cloning is going to eventually produce a boom. I wrote another essay just talking about how this cloning in general is used in agriculture, it's used in microbiology, it's used in all sorts of cell work. We are going to use cloning as a conservation tool for, to deal with the species that are threatened and endangered, to introduce more diversity into the gene pool, etc. Cloning is only going to increase. Cloning technology is only going to improve over time. I think it'll be the first human clone that will really catalyze interest in cloning and really get people to demystify it and just see it as another tool in our tool chest.
Right. And we also wanted to kind of, get your thoughts on synthetic life, because there are these people such as Michael Levin from Tufts Labs, the cutting edge of creating, synthetic beings, right from Xenobots, which are replicating organisms. And then there are also teams who are kind of tinkering around with these things called organoids and building biocomputers. There's one which is Cortical Labs. There's Final Sparks. They're taking this pluripotent stem cell and they're turning it into an organoid, brain organoid, and they're actually building biocomputers. So what's the most exciting synthetic life application that you personally have come across?
Well, I'm gonna push back a little bit. It's not synthetic life. These are people working at the forefront of cell biology and manipulating cellular systems. They're not building them from scratch. Although there's a whole group that do what's called protocells that aspires to be able to build a living cell from component materials that are all defined. These organoids are essentially little clumps of cells, multicellular structures that are more representative of an organ system, whether it's the brain, the heart, skin, muscle, than just a single cell alone. So they're useful in doing things like drug testing and evaluations of multicellular behaviors. Organoids certainly have a place out there, and they're kind of cool. Some of these little bio-robots that they're making, they're toys. They're toys, Xenobots. And they're not fully reprogramming these cells. They're making little clumps that behave in certain ways. And I love it. It's really playful. It's safe. It's not going to take over the world. This type of programming, I think, is necessary because remember, we don't have, I think in terms of fully synthetic cells. And again, you have to realize we're not making the cell. We're only coding the genome of that cell. So it's like taking an existing computer and reformatting the hard drive and loading a new operating system in. Like we're not building a new computer. The cell exists, but we're either recoding or making a completely synthetic genome. For the groups that are doing the fully synthetic genome, there's only been 5 or 6 cells made ever, all of them bacterial species. So we are at the very beginning of this field. Now, there are hundreds of thousands of genomes in the genetic databases already that we've sequenced of various organisms, small ones, single-celled. So we've got plenty of templates to start building on once it's cheap to make a synthetic genome. Again, single-celled genome, about 5 to 10 million base pairs, sometimes less, sometimes more. But, generally the E. coli, which is my cornerstone, is about 5 million base pairs of code. So that's kind of my gold standard. When we can make 5 million base pairs of genetic code with precision and boot it up to make a single-celled organism like E. coli, all of microbiology suddenly goes from a science where we're trying to figure out what it does to engineering, where we can build it with precision and modify that genome with precision in any way that we like. And that's just an economic barrier today, a technical and economic barrier. We've demonstrated we can do it. Proof of concept, done. Craig Venter, back, in 2010. Power to him. More recently, Jason Chin in the UK, who's been making recoded versions of E. coli, changing the low-level coding on E. coli, but basically making new variants of it. Superb work, but again, done by elite scientists. But as the tools for synthesizing and assembling microbial scale genomes comes online, suddenly that power comes to, anyone that has access to the tools. So I think coding biology, coding single-celled organisms is going to mushroom like the way the personal computer did. That being said, because I champion the technology, I champion people learning to do this work. We need to build an education system around this. We need guardrails. And the biggest problem, in my opinion, and again, you'll find essays from me around this, is that we're going to get viruses. Because viruses, we are at a technical point today where anyone that really wants to can go and make a virus. It's not something you can just do online easily today, but the tools are there. It's not that expensive. And it's not hard to go and register a biotech company and start to do this, with some guardrails and for different applications. And I'm a huge champion of writing viruses because that's how we make gene therapies. That's how we make oncolytic therapies. That's how we load programs into cells. They are literally viruses are the biological equivalent of a USB stick. It loads a program into an existing cell. Without a cell, they're inert, just like a USB stick is inert without a computer. But that's how we load programs. So they're always going to be here. But COVID kind of taught us, there's a huge asymmetry between a new virus appearing and our ability to detect and defend against that. Much of my speaking today is related to biological defense and biological sensing and countermeasures, because those have clearly fallen behind the forefront of our ability to make things. We saw the same dynamic with computing, by the way.
Right. Yeah. So I want to get into biological defense, but before that, this interesting AI with biotech convergence, because there's exciting things happening. We have these large language models, large reasoning models, and then we're having foundational models which are being built specifically for biology. There's the famous AlphaFold, folding for protein folding. There's NVIDIA's BioNeMo, and there's so many others. So how do you see this convergence of AI and biotechnology reshaping the future of healthcare and drug discovery? And what new breakthrough or changes should we expect as these technologies mature?
I think it's the best thing to happen in terms of diagnostics and design. Look, a single cell, a simple one, again, I work with bacteria. E. coli makes over 4,500 proteins, give or take. And we can't hold that many variables in our head and understand all the connections between that many components. It really takes a meta system to do that. And that was one of the reasons why I went to Autodesk, because putting together a skyscraper with literally millions of components takes software tools. And building a complex airplane or spaceship requires software tools. Those software tools have lagged in biology, but now they're coming on full bore with AI tools. And that's because they speak these languages natively: DNA, RNA, and protein. The LLMs can speak any language you train them on. They've read all the scientific papers, which is huge. They understand all the stories. They understand the CVs of everyone. That these tools are more than just designers. They can help you design experiments, help you build and operate a company, help you solve economic models and get through regulatory. Like, everyone is using them, from, the beginners all the way to the biggest pharma companies. So they're having a huge effect. And this is one of the things I'm championing right now, just in terms of biosensing, because, for example, DNA sequencing should be really cheap. It's a 50-year-old technology now, and yet no one has a DNA sequencer at home. Well, I shouldn't say it. I know a few people with DNA sequencers at home from Oxford Nanopore, but it's not like toasters or microwaves. Or air fryers. Like, it is still a very elite technology. But when we crack that technical barrier, which we could do in a year, it could be a device in all of our homes where, if you need to swab anything, your nose, your skin, your mouth, whatever, you swab, put it into this device, it'll sequence everything on that swab, and you'll have an AI analyze that information better than any genetic scientist you could call up, and do it near immediately. That technology is something I want in every single home moving forward, because, man, my kids go off to a birthday party, they come back with a fever. What do they have, right? We're completely blind to most of the bacteria and viruses that circulate in our world. And frankly, I've had enough of that blindness. I want to see in K and understand that world around me. And of course, I want to manipulate it too, but it starts with understanding and being able to see.
Right. Yeah. Hopefully, once these DNA sequences, become more cheaper, it gets democratized, and hopefully it should be in everybody's houses. And then we will get into the personalized medicine, unlike what we are doing at this point in time. You spoke about viruses, it's got these pros and cons also, Recently, the researchers, they kind of found these zombie virus, which was thawing in the permafrost in Serbia. Do you think that this is something that we should take seriously as a real world threat? Is it possible to re-engineer, weaponize these zombie virus? Or is there like a greater risk in the increasing accessibility of dangerous biological agents?
Sure. Look, any technology can be weaponized. Any technology. It's all about the use intentions. Now, there's also going to be things that are accidents. Again, if you thaw out ice and release viruses that haven't seen any species for, literally 30 or 40,000 years, maybe longer, there is the potential to seed a new infection that we're naive to and would have to go and make a vaccine for. But nothing that has existed in the past is foreign to us. We're all connected, and there's been a huge amount of co-evolution on this planet. We all run the same code, essentially, every plant, animal, and microbe, which is awe-inspiring to me, but kind of, I can rationalize why that's the case, but it's still pretty awe-inspiring. Anyway, we all exist on the same biological internet. I think the biggest risk today isn't the natural processes. I think it's the fact that now that we can code biology and program it. The risk to me is that some of the smallest agents, biological agents, can be the most dangerous. And again, chief among these are the viruses because they have very small genomes, well within our technical ability to make cost affordably today. They self-replicate using cells as hosts. So they're kind of parasitic and they can spread globally very quickly under the right conditions, which we can model, simulate, and design for if we're designing them with precision. Naturally, they may appear as a pandemic just through the combinatorial engine of nature, rarely. Again, the last pandemic before COVID was 100 years before, with various factors feeding into that, like our ability to move around the world and infectivity and particle count at burst. It's realized it spread out. And so COVID was kind of our wake-up call, our refresher course on pandemics. Whether it was engineered or not is irrelevant to me, other than the fact that we can't easily identify whether it was engineered or not. And that's not okay. But it's certainly getting easier to go and make these agents that could have, if designed to be malicious, could cause huge harm or death, not just to humans, but to any species we target, whether it's an agricultural crop or animal or any species. This to me is my biggest fear, because in all outbreaks, there's a cycle of response and apathy. You know, we charge in to Africa with our Ebola treatments and med teams, and then 2 months later, there's no funding. We go through those cycles, but I think that we have to keep synthetic viruses, designer viruses, really high on our list and figure out some way to watch over each other and monitor each other cooperatively, because it's just so accessible, so cheap, and a single inoculum in a local area could spread globally. Like, we've never had this level of risk before. And I think humanity has to step up to counter that if we want to move into doing more productive things. And this is exactly what we saw with digital computing. When we started digital computing, everyone was friendly, and then we got the first viruses. And I just wrote in a presentation the other day about the brain virus, the first one to start spreading through PCs. It wasn't malicious, but they quickly, these brothers in Pakistan had accidentally created a digital pandemic, and they realized, holy crap. And the world changed. Today, in the cybersecurity world, it's AI systems versus AI systems all the time, which is largely what happens in biology. But now we have to factor in malicious engineered biology.
Right. We're living in such a fantastic time in human history. And yet it's also the most scariest time, because we have democratized these tools, and we've made it accessible. You're someone who champions open source, and I believe that open source should be the way. So my first question is, since you champion open source, what are the pros and cons of making these tools accessible to everyone? That's my first question. And we also live in a very complicated world, and given the risk of the geopolitics, there's always the risk of like a bio warfare kind of kicking off, or a future pandemic caused by maybe like a highly infectious virus. How prepared do you think our current biosecurity and biodefense systems are to respond to such threats? And what are the biggest strengths and gaps in our global readiness today?
Yeah, no, I think COVID was, basically showed that everything we're doing when it comes to monitoring biothreats and countering them, got a failing grade. It's not that we were doing nothing, but they certainly didn't work very well. And we were blind at the beginning, and there was huge amount of confusion. As things started to be better understood and we started to respond to it, and there was certainly a huge lack of cooperation between nations. It was more competitive. So I think we just get a failing grade there. But thankfully, it was more like a bad flu than something with 90% mortality. So when it comes to, they have to be rebuilt. I wrote an essay 3 or 4 months ago titled, provocatively, "The Bioweapons Are Coming," which is, again, just my thesis that whatever happened in digital computing is going to come to digital biology. So we can't be naive. We have to prepare for this. And again, it has to be done cooperatively. Profit isn't the goal here. Actual, meaningful, useful, resilient defense and sensing is. So that's just a challenge moving forward. Will we get through it? Yes. Will it scare some people? Yes, and good. They'll invest in it. Will there be accidents and abuses? Yes, we're human beings. So every single one of us is complicated. And today it doesn't have to just be a nation state that does something like this. It could be an individual that's just got a chip on their shoulders. And I had so much faith that humanity would start to recognize and proactively prepare for this technology pre-COVID. But it was pretty clear during COVID that no, there is so little understanding of these threats, even in defense groups, military groups, that it's a huge problem. And of course, it's going to continue to be a problem moving forward as the nature of conflict continues to change. Today, it's drones. Tomorrow could be viruses, which I look at as nano drones. So who knows? All I know is this is with great power comes great responsibility, and our ability to program life, which has largely emerged over the last 25 years, really forces us to step up as a species. And it's going to be really distressing to some people that are more theological because people say this is like playing God. No, it's the world around us that we've reverse engineered and now understand sufficiently to manipulate, like electricity, like hydrology, like any other technology, except that this is the only technology we did not build, outside of, stars. Like life is the only technology we did not build, outside of the matter that it, populates the universe. And so this is a huge transition for our species to take that responsibility and use it wisely for good.
I really hope that, that happens, because we're the cusp at such fantastic tools or tech which is converging. It's AI converging with biotech. Then there is nanotechnology. Then there's robotics. Then there is artificial intelligence itself, which they're saying that in the next few years could become generally purpose intelligent. Then we are also we possibly might transition from coding with zeros and ones binaries to possibly qubits, like a quantum computer where we'll be actually tinkering around with the source code of life, atom itself. So we are at such a fantastic point of time, but yet I think the human race, we are self-preserving, we're going more towards conflict than collaboration. And I hope that narrative kind of changes, and we understand that we are sitting at a fantastic point of time where we could possibly maybe augment the entire race, and maybe this could be our next evolutionary step. And I hope that we take a conscious step which is collective. And I hope that we don't go towards conflict. Now, you've been part of Autodesk, and you pioneered 3D printing of cancer-fighting viruses and designer organisms. Could you share the latest update on your 3D bioprinting work, and how close do you think we are to seeing functional, 3D printed human organs, because that's something which has been like a dream or a vision.
Yeah, no, the best 3D printer to make an organ is an animal. And you should take a look at the company eGenesis, which is really leading some of this work, making humanized pigs. And they're going into patients now and really pioneering that work. So I don't speak to it that end. The company I co-founded, Human Genomics, I'm just an advisor to today. And really, to get the state of the art of what they're doing, you should interview them. But let's just say, from what I can see...
Actually, sorry for interrupting. I actually did interview the CEO of eGenesis on the podcast.
That's fantastic. Luhan, absolutely amazing work. And we're only going to get better and better at making these systems. I tend to work right down at the lowest level, fusing. I want to see the fusion of silicon-based computing, semiconductors, CMOS-based semiconductors, fusing with biomolecules and or cells. Like Petri dishes, the plastic plates that we use for growing organisms don't have to be dull or dead plastic. They can be sensor systems. Chips can merge. And so we can get real-time communication bidirectionally with biomolecules. These are really exciting. You extrapolate this moving forward, we're going to be able to program cells. We're going to be able to make any protein, any mRNA, any DNA that we want with more and more precision. We're going to get more real-time sensing of the biological world and manipulation of that world. We're going to be able to build things like organ systems and other biomachines moving forward. And I think it's just going to become almost like an exploration that goes as deep and wide as going into the into the micro and the nano, that it's kind of the counterpart to going into space and low Earth orbit, the moon and beyond. We're making those steps down into this world with more and more precision and engineering. And I think it's an unlimited play space. It is, we're going to learn a lot of lessons on the way, but the reasons for doing it are so compelling that there's no stopping it. Kind of like no one could have stopped the evolution of these devices even if they wanted to, saying, I'm not sure we should do this. No, there's just too many compelling reasons to move forward. So there'll be some speed bumps along the way, but that's part of developing any technology. And yes, absolutely, I hope we come together and do this cooperatively rather than competitively. I don't mind economic competition. That's great. It's when we start being violent to each other that we have a huge issue. And today there's David and Goliath. We see this playing out in Ukraine with drones. If people start to be persecuted enough today, they're going to start fighting back with these technologies as well. When you've got nothing to lose and people are pushed into the corner, you get some really bad edge effects. This is why I champion open source. It's the backbone of the internet today. It's not, we build on top of it. All of biology is open source. It's executable code. There's no, we've learned to, it's fully accessible. You can get the genome of any organism very easily. And now with decentralized science starting to emerge and decentralized funding systems, it's no longer centralized, which builds more trust and access. So I think DeSci is kind of the new open source, and with a digital foundation that I think is very powerful.
Right. I think so far, throughout, I think the pharma industry and science itself was extremely centralized, with these new ethos coming in, which is open and decentralized, especially with decentralized science. And I'm sure the entire game is going to completely change in the next few years. Will you be able to give an overview of your new book you co-wrote with Amy Webb, The Genesis Machine, and what future scenarios excites you most and which ones worry?
No, so the book is really a book about this science that's not written for the scientist. It's written so that to be accessible, and it tells stories of some of the key people and players that have built the foundation of this technology. The book is 3 years old. A lot of things have changed since it was written. Some companies have gone away. New technologies have appeared because it's a snapshot, a moment in time. But this story is still being written every day. There's my friend Adrian Wolfson has a new book coming out next year, The Future of Species, that I think will continue to do this type of storytelling. The scenarios Amy and I tell in the book are great. Each represent some of the challenges around people living longer, moving into space, the challenges of connecting the dots to do more effective security, etc., and also dealing with some of the changes in human reproduction and our ability to understand who our future kids might be and manipulate them. So they're meant to be provocative and thought-provoking, but they're not truth. They're just scenarios. And I think that's really illustrative. Like, it's been the science fiction authors who have done most of the storytelling. And I really wish scientists were better at telling their stories of the amazing work that they do to a wider audience. But, I've come to understand that it takes years for these new technologies to kind of permeate into, often it's generational, to permeate in, across the world. And that, when it comes to collective appreciation of something, we're still working at the level of music and chasing balls, and where we can come together and kind of all appreciate something really emotionally and really viscerally. We have no building anything complicated, building big systems. Whether it's like Airbus is generally something where it takes a corporation, often with tens of thousands of people working collectively on parts of that goal. The only time scientists have come together to do a global project was really the Human Genome Project, which is why, the Genome Project currently paused is still such a huge attractor for me, and I think will ultimately be one of the most important things I've ever seeded in my career.
Right. Andrew, since you talk about programming life, there is a certain group of people who say that the world itself is not real, it's simulated. There is this technology called virtual reality where we are building the entire, maybe the physical world though it's extremely rudimentary at this point of time, we talk about something called as a full dive virtual reality, where we would be possibly, maybe in the far out future, we'll be able to kind of code simulate the physical world. Since you talk about programming life itself, what are your thoughts? Because, we've got these trillions of cells, which is run by electricity. How much of us are we truly biological, or is everything code?
The code builds the hardware. It doesn't build the experience. Raise a number of different issues here. Are we living in a simulation? Yes. Your reality is built in a piece of meat, inside your skull. So we live, in some ways we already live in a simulation. We can never model our reality with enough resolution, no matter how much compute. It's just too detailed. You've got to go and experience it. I think virtual reality and these tools that we're building are going to be huge for just being able to visualize these invisible processes that go on inside biological systems and complex systems. I think that's huge. And overall, I think it will be this informational layer that really brings cohesion to all this information that goes into making a single microbe, let alone us. But all this is playing out, and it's the most interesting time to be working in information technology, high performance computing, biosciences, medicine, etc. And I think that's a pretty good place to leave it.
Thank you, Andrew. Really, really appreciate you taking time and being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye-bye. Thank you, Andrew. Really appreciate it.
Thank you so much. I really appreciate it.